Hello, my name is Joshua Mugabi. I welcome you to Proteins. Lesson objectives: amino acid structure, polypeptide formation, level of protein structure, protein functions, denaturation, gene to polypeptide, proteome. Welcome. Amino acids. Proteins are comprised of long chains of recurring monomers called the amino acid. So we are looking at the amino acids, and the amino acids are the monomers that build up a polymer, which is a protein. So a protein is made up of a long chain of recurring amino acids, meaning that it is made up of amino acids joining together as monomers to form a complex compound, which is proteins. So when we look at the structure of the amino acid, I want us to look at the structure of the amino acid. You will see that the amino acid has got an amine group, which has got nitrogen and 2 hydrogen atoms. It has a variable group. The variable group keeps on changing, and then hydrogen. Then it has got a carboxyl group. That is the structure of an amino acid. So when you look at the molecule, this is how the molecule looks like. You can now identify the molecule and look for where you find the R variable group, where you find the amine group, and then the carboxyl group. There are 20 different amino acids which are universal to all living organisms. 20 different amino acids that are universal to all living organisms. Let's look at the types of amino acids. We have amino acids that are non-polar. And among these ones, we have glycine, we have alanine, there is methionine, cytins, we have phenylalanine, leucine, isoleucine, valine, tryptophan, proline. All the other ones were non-polar. Now we have the polar. Polar, we have serine, threonine, triosine, asparagine, glutamine, asparagine. All those ones are polar. Those are the types of amino acids. Now we have the positive charge. Those that have positive charge, we have lysine, arginine, histidine, arginine. Those ones have got a positive charge. Aspartate, glutamate. We look now at the polypeptides. Amino acids are covalently linked together by peptide bonds to form polypeptides. So we are saying that the amino acids are the ones that form the polypeptides, or they are the ones that form the proteins, or we can still say they are the monomers of the proteins. But when you bring a monomer and a monomer, you form a dipeptide. Now, a dipeptide, those two amino acids, they are covalently bonded. They are covalently bonded. And the bond that links the two together is called a peptide bond. So we say that amino acids are covalently linked together by peptide bonds to form what we call a polypeptide. Now, when you come here and you look at our structure here, this is a polypeptide or a dipeptide. And if you cut it in the middle here, the other side we have an amino acid, and this side it is an amino acid. But where we are seeing yellow here, it's where we have our bond, which we are referring to as the peptide bond. We have the peptide bond here. So it is a connection between one amino acid and the other amino acid, and we end up losing a water molecule. So which means that this is a condensation reaction, and they form a covalent bond, which we refer to as a peptide bond. So this one goes through the ribosome. It passes through the ribosome. It's where the amino acids are joined together and we form a polypeptide bond to form the polypeptide, to form peptide bond to form a polypeptide. Polypeptides are synthesized on a ribosome via the process called translation. Now we are preparing the polypeptides. These polypeptides are prepared by the process called translation, and this process of translation takes place on the ribosome. So we shall look at how the polypeptides are formed and how the ribosome forms the polypeptide. But at the moment, we just want to know that when we are forming our polypeptide, the polypeptide is formed by a process called translation, and that one takes place in a ribosome. That's where translation takes place, and the translation is where we are connecting the amino acids together to form a polypeptide. Of course, it takes a condensation reaction because we end up getting a water molecule. The peptide bonds are formed by a condensation reaction. They are formed by a condensation reaction. And what is a condensation reaction? A condensation reaction is what produces a water molecule. So we have already seen that we have one amino acid on one side, and then we have another amino acid on the other side. So when they are passing through the ribosome, that is where we connect one amino acid to another amino acid. And when we connect them, we form a bond which we are calling a peptide bond. But that bond to be formed, we have to release a water molecule. And when we release a water molecule to form a peptide bond, that is what we call the condensation reaction. Let's now look at the protein structure. The protein structure. Proteins or polypeptides fold into unique shapes, which may be essential to their functions. Now we have polypeptides made from amino acids, and they are going to make different shapes according to their functions. So a polypeptide, when it is made according to its function, that is how it is going to fold. So we have different types of polypeptide as we shall see them. We have the primary polypeptide, secondary proteins or polypeptide, tertiary polypeptide, and then quaternary proteins. We are going to look at them each one individually. There are 4 levels of protein structures. There are 4 levels of protein structures. We have the primary, we have the secondary, tertiary, and quaternary. As we have already seen them, we have talked about them here. This one is a primary protein. This one is a secondary structure protein. This one is a tertiary structure. And the other one, we have the quaternary structure. The amino acids are linked together by condensation to form a polypeptide. We have moved away from this side where we say that the amino acids, they come together or they are linked together through a process called condensation reaction to form a polypeptide. So from one amino acid to another, we have a bond which is formed. That one is the one we are referring to as the condensation. For example, we have a lysine here, then there is also lysine here. There are different amino acids here. Now those different amino acids, they are joined together by a peptide bond. And when they join together by peptide bonds, that is now when we form our polypeptide. Now this one is our primary structure where we have the amino acid linked together by a condensation. So when you link this amino acid to this one, you are going to lose a water molecule. If you link that one to this one, you lose a water molecule. Link that one to this one, we lose a water molecule until when you finish the last one. So that releasing of water is what we are referring to as the condensation. So from simple amino acid, another amino acid, then we end up forming polypeptides. Now let's look at the primary structure. The primary structure describes the order of amino acids in a polypeptide sequence. Now, we have a primary structure and we are saying this one is just describing the order in which the amino acids are arranging themselves in a polypeptide sequence. Now, if you look at this structure which we have here, we have this one. This is our polypeptide. And the ones that we are having as these small dots, the circles that you are seeing, those are amino acids. Those amino acids are joined together. And when they are joined together, they form a structure which is linear. And that structure is the one that we call a primary structure. It is a polypeptide. And a primary structure is just describing the order of amino acid in a polypeptide. This is a polypeptide, and it is made up of amino acids arranged together. We say that there are 20 amino acids. There are 20 amino acids with distinctive chemical properties, in all living organisms. It is formed by covalent peptide bonds. Between adjacent amino acid. We have already talked about that one. We have looked at the circles being amino acids. And those amino acids, every amino acid to another amino acid, there is a peptide bond. And that peptide bond is the one which is joining one amino acid to another amino acid. And when all of them are connected together, now we form our polypeptide chain. but those amino acids are connected to each other by a covalent peptide bond, and that covalent peptide bond is formed through the process of condensation, where we lose a water molecule. So here is our amino acid, and this amino acid, one side will connect one amino acid, and then the other side we shall also connect another amino acid until we form a long chain, which we are calling a polypeptide. The primary structure still, it controls all subsequent levels of protein structure. So the first structure that we start with is the primary structure. And this one is the one which is going to control all the subsequent level or structures of the protein that are coming next. We say that we have the primary structure, we have the secondary structure, tertiary structure, and quaternary structure. So the structure we start with is the primary structure, and it is the one which will determine the secondary structure. It will control the tertiary structure and the quaternary structure. So the beginning structure from amino acid, we shall have a primary protein or primary structure. It's the one which we are going to have first. The amino acids can be linked together in a sequence giving a huge range of possible polypeptide. So we are saying the amino acid can be linked together in any sequence giving a huge of possibility of polypeptide. Now we say we are having 20 amino acids. There are 20 amino acids with distinctive chemical properties. Now these 20 amino acids, they are going to be arranged differently. They will be arranged differently, and that one, the arrangement will keep on changing. That changing of the arrangement is the one that is going to give the different structures of the different proteins that we form because of the different arrangement of the amino acids. So you can say that there are only 20 amino acids. How do they form different types of protein? Of course, when they are forming the different types of protein, these amino acids, the way they are arranged is different. One amino acid may come and then another one and another one. And when you look at another protein, the arrangement of those amino acids may be different. And if we have different arrangement of amino acids, that one gives us a different polypeptide structure. So that's why we are saying amino acids can be linked together in any sequence. They can be linked together in any sequence, giving a huge of possible polypeptides. Let's look now at the secondary structure. Secondary structure describes the folding of a chain into repeating arrangement. I want us now to understand this one very clearly. We are looking at the secondary structure, and this one describes the folding of a chain, into repeating arrangement, You remember when we had the primary structure? The primary structure was just a line, a linear structure. Now, when you bring the primary structure and you start folding it into chains, and you keep on moving, folding, then you get a secondary structure. So when you look at our structure here, the one we have, a secondary structure, it comes from here, it comes like this, and then it folds. You can see it folding, it keeps on folding, it keeps on folding and folding like that. Now that folding makes it to move away from being a primary structure to being a secondary structure. So that's why we're saying a secondary structure describes the folding of a chain into a repeating arrangements. So we have two types of folding. We have one that we are calling the alpha helix. The alpha helix is the one which has an alpha helices. They are just coiling around, okay? Coiling around, like the way you are seeing them. Those are alpha helix. Then we have the beta pleated sheet. The beta pleated sheet. These ones, they come and they are just twisting. Twisting, they are folding, but twisting around. If you look at this one here, it twists and then it twists. The way even the arrows are showing you, it is twisting in such a certain way. Now that one is what we are referring to as the beta pleated sheets. Those ones are called the beta sheets. The other one, alpha helix. Now, all of them, they are secondary structures. They are secondary structure. But it is going to depend on the type of the sheet. Is it a beta sheet or an alpha helix? But they are all folding the primary chain to form the secondary structure. Still, it is formed by stabilizing hydrogen bonds between non-adjacent amino acids. The secondary structure is formed by stabilizing the hydrogen bonds between non-adjacent amino acids. I want you now to understand this, okay? Now, if you look at this structure here, I want you to see this structure here. That is a primary structure. You see this second structure here. That is also a primary structure. But when they come and fold like this, now when they are folding, their hydrogen bonds, they will form hydrogen bonds between this structure and this structure. They will try to form hydrogen bonds to join themselves here and here. Those are the ones that we are calling the hydrogen bonds. And these hydrogen bonds are between non-adjacent amino acid. So this hydrogen bond, which is formed here, going here, there is an amino acid here. And here also, there is an amino acid here on another structure of a protein or a polypeptide. Now, the structure here and the amino acid here and the amino acid here, they form an attachment through a hydrogen bond. But these amino acids are not adjacent to each other. That's why we're calling non-adjacent amino acids. So they are on different molecules, different amino acids, different polypeptides, but forming adjacent amino acids. So that is what we call a stabilizing hydrogen bonds between non-adjacent amino acid. And that one is in a secondary structure. I hope you have understood that one. So you have a structure here. This is a polypeptide. When it comes, it is going to fold, and when it folds, and then here, now there is another polypeptide. Now there is going to be an amino acid here. There is an amino acid here, amino acid, amino acid, amino acid in that polypeptide. And here also there is an amino acid, amino acid, amino acid, amino acid. Now those amino acid, when they come and form bonds between themselves, this one and that one, and then this one forms a hydrogen bond with that one. That is what we are calling stabilizing the hydrogen bonds between non-adjacent amino acid. This amino acid here is different from this amino acid. So when this amino acid here forms a bond with this amino acid, they are not adjacent amino acids because they are on different polypeptides. So that's what we are saying. In secondary structure, there is stabilizing of hydrogen bond between the non-adjacent amino acids. Sequences may form alpha helix or beta pleated sheets or form random coils. So we are forming the different coils. I told you this one here, we have the alpha helix. The alpha helix is this one here, which we are having here. They are just rotating around. And then we have the pleated beta sheets. These ones are rotating, but they are twisting. They rotate, but again, they do the twisting. They are the ones that we are referring to as the beta sheets. And these ones that are just rotating around, we refer to them as the alpha helices. So they may form random coils. They may form random coils. The coils may be random, like the way you are seeing this one. Coming from here, it comes here, and then forms another coil going up like this, and then it forms another coil, then it comes this side like this. So they may just form a random coil. Well, as you're seeing this one, it comes here like this, then it goes like this, and then it goes up like that. So they can just form what we are referring to as a random coils. They are not specific coils that it is going to go this side. They can form random coils. So those are the sequences. We have the alpha helix, and then we have the beta pleated sheets. But all of them, they are secondary structures of proteins. Then now we go to the tertiary structure. We move to the tertiary structure. Now the tertiary structure describes a way or the way a chain folds into three-dimensional shapes. It describes a structure in which a chain is folded into three-dimensional shapes. So here, the structure is forming, okay? What we are referring to as a tertiary structure, you can see it is forming in a three-dimension shape. It can come and then this direction, this one is coming this direction, the other one is coming going the other direction, and then the other one is moving in this direction. All of them, they are twisting around. Now that one is what we are referring to as a three-dimension. When you look at this one, it is going down, and then others are going up, and then we have these ones that are going on the side. So that one is also a three-dimension structure. So you move, when you see it forming three-dimension shapes, then you know that now that one is a tertiary or a tertiary structure. You can look at it and see this one is going downwards. And then you can see this one is a little bit horizontal. Then you can see this one is diagonal. So those ones are the ones that we are calling the three-dimension shape. So we can have this one, which has a structure, roll, and insoluble in water. This is the fibrous protein. The fibrous protein, it has got a structural roll. It has a structural roll because it is insoluble in water. Then we have a globular protein. The globular protein is the one which is coiling around, and this one, it has a function role because it is also soluble in water. So you have the fibrous protein. You can see how the fibrous protein is folding. It is folding like as if they are linear, folding each other, forming a rope. And then when you look at the globular protein, for it, it is just folding in any direction. Globular, it is soluble in water. Fibrous, it is insoluble in water. The tertiary structure is formed by interactions between variable groups. It is formed by interaction between variable groups. They can be ionic bonds, they can be hydrogen bonds, and very many others. So you look at this one, they are going to interact in very many ways. This amino acid may interact with this one here. This one may interact with the other one here. This one may interact with this one. This one may interact with the other one. So that's why we are saying that it is formed by interaction between variable groups. There are very many variable groups which are interacting with each other. They can be ionic bonds, they can form hydrogen bonds, and very many others. And in tertiary structure, affinity and repulsion between the different sides chain will affect the overall folding. So when you look at this folding here in the tertiary, the way they are folding, it is going to depend on the affinity and repulsion between the different sides, different side chains. This one is the one which is affecting the overall folding. So the way it is folding, you may find that if you look at this one, it is folding like this and folding like this, then going upwards and then changing direction coming this side. Why is it folding like that? It's because at the adjacent amino acid here and here, you may find these amino acids here, they are repelling each other. That's why they are not very close to each other. Whereas you find that some of them, they are very close to each other. Some are very close, but others are very far away from each other. Where you are seeing very far away from each other, you just know that there is repulsion at that particular point. And where you see them coming very close to each other, that means there is an affinity. They are attracting each other. Some of the molecules there, they are attracting each other. So this attraction and repulsion is the one which is going to determine the overall folding of the structure, depending on the different sides chain, how they are interacting with those ones next to them. They can be repulsive or they can be having affinity for each other. Now, still we are saying the amino sequence determines the three-dimensional conformation in the protein. So we are saying that this amino acid sequence is the one which is going to determine the three-dimensional conformation of the protein. How are we arranging the amino acid? If you have different amino acids arranging themselves differently, those ones are going to determine the conformation of the structure, how the structure is going to be. So when the structure rotates around and it has different conformations of amino acid, it is going to determine how the folding is determined. Either they will attract each other or they are going to repel each other. So when these amino acids are facing each other, some of them may have affinity for each other. Then they will attract each other. And when they attract each other, then that bond, these two polypeptides are going to be very close. But whereby you find that they are repelling each other, there is going to be a gap between the two polypeptides. So that's what we are saying, that the amino acid sequence, how these amino acids have been arranged, is going to determine the three-dimensional confrontation, how they are arranging themselves in the protein. It will be determined by the sequence in which the amino acids were arranged. Now we move to the quaternary structure. Let's go to the quaternary structure. Now, a quaternary structure describes the presence of more than one polypeptides. Here we have more than one polypeptide chains, or the presence of additional prosthetic group in a biological active protein. So in a quaternary structure, we are having different polypeptide chains. They are different polypeptide chains which come and join together and they form what we are calling a quaternary structure. Or sometimes there might be a prosthetic group. There might be what we are referring to as a prosthetic group. This is a structure which is not part of the protein. For example, if we look now at our structure here, the quaternary structure here, when you look at it, it has got very many polypeptide chains. There is this type of polypeptide chain. There is this type of polypeptide chain. Then on the other side, we have another polypeptide chain, this one. And then we have this other type of polypeptide chains. Those, all of them, they are coming together. And then we are having what we are calling the prosthetic groups. If you can see that structure in the middle, which is like a coin there, that is the prosthetic groups. Now they are coming together. There is four, if you look at them, here we are having 4 polypeptides. This is one, this is a second, we have a third one here, we have the 4th one, the other side here, the 4th one here. So which means that we have 4 polypeptide chains joined together. And those 4 polypeptide chains, now we also have 4 prosthetic groups inside, 1, 2, 3, and 4. Now that structure, when it comes together, now that structure, we call it a quaternary structure. Because it has got this one, this structure which we are having here, all this structure which we are having here, it is a tertiary structure. This one here is a tertiary structure. The other one behind is a tertiary structure. This one, this side, is also a tertiary structure. So we are having 4 tertiary structures coming together and forming one structure. And that one structure which is formed is the one we are calling a quaternary structure. Many polypeptides and the prosthetic groups coming together and forming one structure. So now that one we can very sure now know the quaternary structure, the tertiary structure, and the secondary structure, and the primary structure. The protein may consist of a single polypeptide or more than one polypeptide linked together. They might be a single polypeptide. For example, here we have 2 polypeptides, types of polypeptides. There is this one here. This one is the same as this one here. They are the same type of polypeptide, but they are joining together. And then you have another one. This one which you are seeing outside there, like that, is the same polypeptide as this one. So we are having 2 identical polypeptides joining together. So that's why we are saying it might be a single polypeptide or more than one polypeptide linked together. This is a polypeptide, this is a polypeptide, but they are the same, but they are more than one. They are joining together. And the other one, they are the same, but they are joining together, 4 of them, and then they are forming what we are calling a quaternary structure. Of course, plus our prosthetic groups. Now we want to look at the level of protein structures. Let's look at the levels of protein structure. How do they arrange themselves? How do we know that this one is a primer, this one secondary, tertiary, quaternary? How can we identify them? Now, we know that living things synthesize many different proteins. Living things will synthesize very many different proteins and with a wide range of functions. So because there are very many functions in a living organisms, and all these functions are different from each other, therefore the proteins which are involved are also different. Therefore these organisms are going to produce very many different proteins. Therefore we shall have different functions. For example, we want to look at the primary or order of amino acid sequence in primary proteins. Now when you look at the primary protein, That's how you will see it. It has very many, they come together, very many of them join together. The amino acids, they are moving along with each other and they are forming a different structure. That one is a primary protein. That one is a secondary protein. You can see that for it, it is forming what we are calling the helix, alpha helix, and then also beta pleated sheets. It has alpha helix and beta pleated sheets. If you can look at them, they are different from the primary. Primary, you saw that they are just linear amino acid joining to another amino acid. Now here you have a polypeptide twisting around, forming an helix, and then also bending around to form a beta pleated sheet. That is a secondary structure. Now that one is a 3-dimensional overall. It is a 3-dimensional overall structure, and it is a tertiary structure. That's how you can see it. It looks like that. It is a tertiary structure and overall. Then we have now what we are referring to as a quaternary. Quaternary, you can see that it is having very many tertiary structures joining together and they form one structure which we are calling a quaternary. That one is what we are referring to as the quaternary structure. Let's now look at the protein functions. What are the protein functions? Now, proteins are diverse. Proteins are diverse, and a diverse class of compounds that serve a number of different roles. We have very many classes of proteins, and they are performing different roles. So we want to see those roles that are performed by the proteins. The first one is the structure. They form structures. For example, we have collagen, we have spider silk. All those ones are structure proteins. Then there are others which function as hormones. For example, we have insulin and glucagon and many other hormones. They are hormonal function. Then we have those ones that are their function is immunity, like immunoglobulin. That one is a protein which is for immunity. Then we have others for transport. For example, like hemoglobin. That is a protein which is its function is to transport oxygen. Then we have sensation. In sensation, we have example like the rhodopsin. That is the cells that are found in the eye for sight. We have other proteins which are involved in movement. For example, in the muscles, we have the actin fibers and the myosin fibers. Those are for locomotion. Then we have others that form enzymes. These ones we have, for example, Lubisco, we have catalase, amylase, and very many other enzymes. So that one is also another function of proteins. Now we look at denaturation. Denaturation is a change in the protein structure. Denaturation is a change in a protein structure that results in a loss. It can be permanent of its biological properties. So we are saying that denaturation is a change in the protein structure that results in a loss of its biological property. For example, the loss of a tertiary structure equals to loss of biological activity. Let us look here now here. It's this one that we are seeing here. This one is a tertiary protein. When you look at it, it is a tertiary protein because it has 3-dimensional arrangements. It is having 3-dimensional arrangement. Now, If it is folding like that, and its functions are determined by the way it folds, its function is determined by the way it is folding. That is how it is folding, and that's how it is supposed to be when it is functioning. Now, if it is denatured, that means that the folding is going to change. Now you can see that it is no longer folded. You can see the bonds that were here, or the bonds that were forming, making it to fold, all of them have been lost because it has been denatured. Now, if it is denatured and the bonds are broken, it is going to unfold. It is going to unfold. It is going to open. And when it opens, that means that now it can no longer perform its function. So we say that the protein has been denatured. It has changed its structure. Therefore, it has changed its biological properties. It can no longer now do its function. Still under denaturation, temperature is one of the factors that can disrupt the bonds. Thermal energy, that is the thermal energy, may disrupt the bonds and that holds the protein together. We have seen that there is a way the protein, the tertiary structure, is folding itself and holding itself together. But when you increase the temperature, that temperature is going to destroy some of the bonds, and when it destroys some of the bonds, therefore the protein is no longer linking to each other. Therefore, it is going to be denatured. pH may alter the charge. If there is pH change, this one may alter the charge of the protein, thus changing its solubility and shape. Some other conditions that may, apart from temperature, that may change the conformation of a protein, it can be the pH. For example, here we have the neutral structure, which we are calling the zwitterion. This one is a zwitterion. It is neutral. It is neutral because it has a negative charge this side and it has a positive charge the other side. So it becomes neutral. Now, this one, when you add acids, you may end up both of the groups being protonated. Here you have a positive, and again you come this side, you also have a positive because you have added a hydrogen ion here. Now that one is becoming positive. Both groups are protonated. We have the amine group and the carboxylic group. Both of them have been protonated. And when you come to the other side, if you subtract, you remove a hydrogen, you will find that here now this one is negatively charged and this one has no charge. Therefore, both groups now we say have been deprotonated. And now we are getting an overall negative charge. So that one, when it happens and you alter the charge of the protein, that one is going to change its solubility and it will also change its shape because we have changed the charges. We have altered the charges. The normal one is what we refer to as the zwitterion, whereby it has a negative charge on the carboxyl group and a positive charge on the amine group. That one is the normal. So if you remove the normal, that means that it is going now to be changing its solubility and also changing its shape. Still under denaturation, amino acids are zwitterion. Amino acids are zwitterion with a negative and a positive charge. They are zwitterion, they have a negative and a positively charged region. So that's how it is. They are supposed to be like that. That one we call it a neutral charge because they have a positive and a negative. Let us now look at gene polypeptides. Now we know that a gene is a sequence of DNA that codes for a polypeptide. A gene, we call it is a sequence of DNA that codes for a protein. A gene is just the way DNA is arranged, and the arrangement of the DNA is the one that has chemical instruction that is going to form a polypeptide. So that arrangement of the DNA is what we refer to as a gene. So this occurs via two main processes. It occurs via two main processes. One is transcription. It is transcription. Now we are going to code for a protein. We are going to produce a protein. There has to be two processes. And the first one is transcription. Transcription, it is where we form messenger RNA transcript that are made from a DNA template. Now the messenger RNA transcript is the one that is going to copy the codes, the information from the DNA, and it will take it in the cytoplasm so that it is duplicated or replicated, or forming copies of that particular expression of DNA or the genes on the DNA. So we have transcription. Transcription, we are just making a copy of messenger RNA on a DNA template. We form it from a DNA template. Now there is another stage which we call the translation. Now in the translation, the messenger RNA came with the information from the DNA. So it has to hand over this information for the amino acid sequences so that they can encode the amino acid sequence. They can arrange the amino acid how they are supposed to be arranged in a particular protein that has to be produced, because the information has been got from the genes that are on the DNA in the nucleus. Typically, one gene codes for one polypeptide. One gene codes for one peptide. However, alternative splicing can produce multiple variety. If one gene codes for one protein, but changing a slight change in the codes can cause a production of multiple variety. A slight change in just one code, it will produce a variety of polypeptides. Some genes don't get translated. Some genes, they don't get translated. For example, the transfer RNA genes. Those ones are not translated. Then the amino acid sequence of a polypeptide is coded for by a gene. So the product that we are getting as polypeptide, it is a gene that gives instruction for these amino acids to be arranged in a particular way so that they can form a particular polypeptide. So the amino acid sequence of a polypeptide is coded by a gene. A gene is the one that gives instruction how the amino acids will arrange themselves. Let's now talk about protein. Protein proteome. Proteome, this is the totality of the proteins expressed within a cell, a tissue, or an organism. Proteome is the totality of the proteins expressed within a cell, tissue, or organism. The total number of proteins that are expressed in the cell is what we refer to as protein. Now, individuals have unique proteome, which may change with the specific conditions. So the types of proteins that are expressed in a particular cell or an organism may change with the change in specific conditions. And the individuals have got unique proteome. Proteome may be significantly larger than the available number of genes. The proteome, total number of protein expression, may be larger than the available number of genes. What brings about this one? We shall see that one. But it is possible that the total expression of proteins in a cell may be more than the number of genes that are available. Alternative splicing may create multiple proteins variants from a single gene. Splicing, alternative splicing, alternative changing, a little change, a very small change in a single gene can cause a creation of multiple protein variants. We say that a protein, when it is being synthesized, the type of protein is going to depend on the arrangement of the amino acid. We know we have 20 amino acids and we have thousands of proteins, but all of them, they are coming from the 20 amino acids. So the polypeptides that we are getting are going to be different depending on the arrangement of the amino acid. So a slice splicing may cause multiple protein being produced from a single gene. So post-translation modification may promote further variation of a protein. That is post-translation modification. If translation has taken place and then there is another post-translation, that is going to cause some little changes, and this one may promote further variation in the proteins that have been produced. For example, phosphorylation of kinases. If there is phosphorylation of kinases, that one may bring about variation in the proteins. Cleavage of zymogen. Cleavage of zymogen. That one may cause a variation in the protein, and very many others. The human proteome is concurrently predicted to consist roughly of 100,000 proteins. So our proteome is currently predicted to consist of roughly 100,000 proteins. How many amino acids do we have? We have 20 amino acids. And now how many protein is currently predicted? It is 100,000 proteins. So you can see. So each individual has a unique proteome. Each individual has a unique proteome. Thank you very much for participating in the protein. We have looked at the amino acid structures. We have looked at the polypeptide chains. We have looked at the level of protein structures. We have looked at the protein functions, denaturation, gene to polypeptides, and a protein. Thank you. Don't forget to subscribe. Bye.